afternoon, everyone. My name is Sarai Hutchinson, and my mentor is Dr. Renata Plattenberg. Today, I'll be presenting my research for the summer entitled Training Acoustic Analysis Software to Identify Frogs Using Machine Learning. My research question is, can we use machine learning to identify frog calls? And my hypothesis is that there is no difference between manual identification and the classifier generated from Kaleidoscope. So what is Kaleidoscope? Kaleidoscope is an acoustic analysis software that uses sophisticated pattern recognition software to automatically scan large quantities of audio files, detecting similar vocalizations and quickly sorting them into groups of similar sounds called clusters. Here's an example of what my Kaleidoscope software looks like. It uses something called the principal component analysis, which is a dimensionality reduction method that is often used to reduce the dimensionality of large datasets by transforming a large set of variables into a smaller one, which contains most of the information in the large set. Smaller datasets are easier to explore and visualize and make analyzing data much easier and faster for machine learning algorithms without having many variables to process. To sum it all up, PCA is simple. It reduces the number of variables of a dataset while preserving as much information as possible. So we take this set of bird sounds and Kaleidoscope would cluster these vocalizations and turn them into groups of these different species of birds. My methods were to obtain the recordings from the Great VI Frog Count Citizen Science Project, and then I would cluster my vocalizations using Kaleidoscope from a reference set of recordings according to the similarity of sound pattern, frequency, which is frequency range, duration, and the acoustic shape of the vocalization. I then manually ID'd a few of my acoustic signals of interest, and this would be used as the training. This would be the training that my machine would get. And then I refined my clusters 2 times. So I took my initial set of recordings and I reran them on this edited cluster that I got. And then I compared my auto ID output with my reference data, which is the spreadsheet that contains the confirmed presence or absence of my 4 species of frogs that I'll be using in my research. So here I'll be showing you the frogs that I used, as well as a sonogram of how the frog sound looked, the vocalization. I'll play sound of a red-eyed coqui, have a whistling coqui, that sounds like this. We have a white-lipped frog, sounds like this, and a Puerto Rican coqui. This brings me to my results. So on my left, it has a comparative bar chart between the present and detected frog species in the acoustic recordings. The present species are those that are found in my reference spreadsheet, and the detected were the ones that were generated by my classifier. So this shows that there was a large number of false positives that were seen across all 4 species of my frogs. And then this would lead to an overestimation of the presence or absence of the presence of frogs in my different recordings at different places. And so on my right, I have a comparison of my precision and accuracy of my 4 frog species. So precision is how close my results are to each other, while accuracy is how close a measurement that I have is to a known or accepted value. My accuracy would be in relation to my reference spreadsheet. And so it's showing that my antilensis had a higher precision than the other three species of frogs, and had an equal amount and approximately same accuracy. While for the leptoalbileverus, it had a very low precision, but a little higher accuracy than there was for the antilensis. And so I performed a chi-square statistical analysis, and my p-value turned out to be less than 0.00001, and this result is significant at p being less than 0.5. And this means that there was a difference in my classifier generated by Kaleidoscope and the reference set of data. And this shows that my classifier did not perform as well as I would have liked it to. And then it showed that my hypothesis is then rejected because there is a difference between my classifier generated by Kaleidoscope and my manual identification, or my reference to the data. And it's showing that my classifier that was generated for E. antilensis was equally accurate and precise, but for albileverus, it had the highest accuracy of the four. My future work then is to revise and refine my cluster, and in hopes of improving my accuracy and precision. And then I would be able to answer what the differences are in species distribution and assemblies across elevational and east-west gradients, different levels of urbanization, and invasive species presence or absence. I would like to acknowledge that this project was made possible by the ECS Honors Fund and my mentor, Dr. Renata Plattenberg. Thank you for listening. Have a great day.